Good day, everybody. I am Matteo, an Italian PhD student working at the Institute of Molecular Virology in Ulm, southern Germany, and I give you my warm welcome to my presentation entitled Corona and Viruses for Dummies. The idea behind this presentation arose a couple of weeks ago when I was asked to prepare a very short presentation for a broad Italian audience to share some basics knowledge about virology and viruses, and especially the new coronavirus, which is leading the pandemic we are all experiencing in these days. Um, I was asked to do this by an Italian friend, not only because Italy has been severely hit by this coronavirus pandemic, but especially because I witnessed a lot of misuse, abuse, misunderstanding, and really, I would say, lack of really basic information about viruses. And this will also be the goal of this presentation, will be to give you guys, let's say, to give a more international audience, really the most basic concepts about viruses and virology. And as such, I would like to start this presentation with a very basic question, which is how could we define an organism as a virus? The word virus comes from Latin and it has been officially translated at the very beginning as secretion. But throughout history, the term virus acquired a more negative meaning, and this explains why nowadays it is very often found translated as poison or poisonous substance or poisonous secretion, as I wrote here. Um, Talking strictly in biological terms, the most efficient way that we could use to define viruses is to say that they are obligate intracellular parasites. And what does this expression mean? Um, Parasitism is one type of interaction between two organisms when, very simply, one lives at the expense of the other. And while the one in this case is the virus, the other is called the host. And obligate intracellular because virtually any virus needs to stay inside of its host cell in order to behave as a virus, so as a parasite. You can take any viral particle that stays outside of its host cell, and this viral particle is completely harmless. Now, the most careful, if not the most picky of you, may have heard the word lives, which leads me to the follow-up question, which to some of you may sound even a bit philosophical. So, could we consider viruses as living beings? Um, so, in my personal opinion, the answer is not. We cannot consider viruses as living beings. And in this light, I would like to briefly justify my thinking. Now, if we could highlight the main properties that are featured in all living beings, from bacteria to humans, first and foremost is the ability of living beings to encode all the information required for living. And this let's say ensemble of information is called the genome. So all the living beings do have their specific genome containing all this information, and this information is very prone to changes, and these changes can be influenced by external factors, and these changes are then transmitted, are transmitted to the subsequent generation, and this is what allows living beings to evolve. Another feature is that any living being can reproduce itself, meaning that it can give rise to another individual. And I would like to underline at this point how this process occurs in an autonomous way. It may be taken for granted, but one feature of living beings is their ability to grow and develop. And in this case, just think at what happens to a human being from the fertilization to the birth and growth afterwards. Um, Living beings do have their own metabolism, meaning that they can carry out a whole series of chemical reactions whose products are essential for the survival of the cells. And finally, all living beings are sensitive to external stimuli and can react to this in different ways. Now, on the other hand, it's important to say that viruses only share a few of these properties. They too encode all the information needed for their replication within a genome, and these information, for reasons that will come up later again, are constantly prone to changes, meaning that viruses as well do evolve. And lastly, viruses replicate, but as I've mentioned in the previous slide, they need to be within a host cell in order to do so. And unlike living beings, viruses do not grow, so a small virus will always give rise to other small viruses. They are entirely dependent on the host metabolism, and they do not respond to external stimuli. Due to these shortcomings, viruses are generally not included in the realm of living beings, and this is why I also support these theories. Still, the question that should pop up in your mind now at this point is, but if viruses are not living beings, then how can they do all what they manage to do? So, for instance, how can this coronavirus be responsible for such this enormous pandemic which is going on at the moment? The, the simplest answer to this question lies in the title of this slide. Both living beings and viruses need to synthesize proteins. And here I will try to explain briefly how this process is regulated. Um, the genome of all living beings, so again, the totality of information needed for life and survival, consists of a molecule of DNA. And DNA is the acronym of deoxyribonucleic acid. And in all living beings, DNA is, let's say, present in cells as a double-stranded molecule, just yeah, like a two strings which are wrapped together. And its information is spelled in a very peculiar alphabet, which consists just of four characters, which are A, C, G, and D. Imagine just a huge stretch of these four characters. For the people who are watching this video who are not really, let's say, expert in biology or in the field, let's imagine that DNA is like a huge instruction manual in which everything that cells have to do in order to live is written. However, this information contained in this manual is not linear. It's not like a book, let's say, but it is all fragmented and scattered. And whenever a cell needs to receive an instruction from the DNA, this piece of information needs to be, let's say, rearranged into something that makes more sense. And this something is called messenger RNA, mRNA in short, where RNA is the acronym of ribonucleic acid. And the process leading to the synthesis of this messenger RNA is called transcription. Transcription because the alphabet with which cells do reorder this information, the instruction contained in the DNA, is also formed by four characters. In turn, the information contained in the messenger RNA must then be translated into the alphabet of amino acids, which unlike DNA and RNA consists of as many as 20 characters. And amino acids represents the building blocks that will make up proteins. Now, again, to make everything simple, if we imagine a cell as a very complex and structured and ordered society, proteins can be seen as the workers that, by cooperating and interacting with each other, they ensure the proper and correct functioning of this molecular society. As endocellular parasites, deprived of their own metabolism, viruses rely entirely on the host cell for translating their information into viral proteins. What strongly distinguishes viruses from living beings is the way that through which viruses encode their information, which could be DNA, either DNA or RNA, and the different strategies that viruses have evolved in order to reach to reach the synthesis of messenger RNAs. And to simplify our lives, so the life of virologists, a guy called David Baltimore came up with a very brilliant and elegant idea of grouping viruses according to the strategies that viruses have evolved to make mRNAs into seven large classes. And this system is called the Baltimore classification. In the first class, there are viruses that mostly resemble living beings because they encode their information within a double-stranded DNA molecule. And these viruses include, for instance, herpes simplex virus type 1, a virus which normally, let's say, sleeps into your nerves, and then when you are, let's say, as stressed as a PhD student, for instance, this virus can get reactivated and cause the labial fever. We have the human cytomegalovirus, which is a huge problem in immunocompromised patients, varicella zoster, the oncogenic papillomavirus, and the pox virus, I think the first virus that it was defeated thanks to vaccinations. In the second class of Baltimore's classification, there are the viruses that have a DNA which only contain one strand. And in order to synthesize the messenger RNAs, these viruses convert their single-stranded DNA into a double-stranded molecule as an intermediate step. And to this class belongs the parvovirus B19, which is responsible for the fifth disease in children. In the third class, we find viruses whose genome consists of a double-stranded RNA. These viruses behave in a similar way to those of the first class, but here the big difference is that the synthesis of messenger RNAs occurs from other RNA. So this is already very different from what happens in living beings. And a virus that affects humans, which belongs to this class, is the rotavirus, so called because at the microscope the virion resembles a wheel, and this virus causes a severe diarrhea in children. In the fourth class, there are viruses whose information is encoded in a single-stranded RNA molecule that is defined of positive polarity. What does it mean? Having a positive polarity RNA means that this viral RNA is immediately translated into proteins when released into the infected cell. Then some viral proteins are responsible for the synthesis of another viral RNA, which is defined of negative polarity. And this in turn becomes the template that is used to produce additional positive polarity RNAs, which in turn they will be translated into proteins, or they will become the new genome for the forming viral particles. In this class, we find viruses like the coronavirus, like the viruses of hepatitis A and hepatitis C, the rhinovirus, one of the many viruses responsible for the cold, as well as dengue and Zika viruses. Vice versa, in the fifth class, there are viruses carrying a single-stranded RNA with a negative polarity. And this RNA is then used to synthesize the messenger RNAs, and again an intermediate RNA, which will then become the template for the synthesis of novel RNA with negative polarity for the new genomes. And in this class belongs, I think, the viruses that most of you, if not all of you, have heard about, like Ebola virus, measles, influenza, mumps, and rabies. The fifth class contains some very peculiar viruses, which they also encode their information in a single-stranded RNA molecule with a positive polarity, like the viruses of the fourth class. But the, let's say, unique aspect of these viruses is that their RNA is not immediately translated into proteins. On the contrary, these viruses do something that is unique and that really no other living being does. Namely, they convert this RNA into a double-stranded DNA molecule, which then becomes the template for the production and synthesis of new viral mRNAs. And the most famous virus that belongs to this class is HIV, the human immunodeficiency virus. In the last class, we find hepatitis B virus. Hepatitis B virus has a double-stranded DNA genome, which, unlike the viruses of the first class, is described as being partially incomplete, because the strand with the positive polarity has some gaps in it. And what happens is that when the virus enters into liver cells, the virus repairs its DNA and then uses this DNA as a template for the synthesis of viral mRNAs. Additionally, hepatitis B does a rather strange thing at some point. It synthesizes an RNA which is called the pregenomic RNA, which is like an mRNA which spans across the entire length of the viral DNA. And from this RNA, the virus does the same thing that HIV does. So it converts this RNA into a double-stranded DNA molecule, which is, however, incomplete. And within the very same cell, this new DNA molecule can either become the genome of the new viral particles, or it can be repaired and start again a new cycle from scratch. I know this is complicated, but trust me, if you got this, then you are, let's say, halfway through to become a real virologist. Now, before spending a few words about the SARS coronavirus, I'd still like to introduce some linked concepts that I think may facilitate the understanding of the coming slides. And these are the concepts of evolution, spillover, and adaptation. Let's start from the basic concept that every living organism on this planet is vulnerable to infection to at least one virus, which here is indicated by the three silhouettes with the different colors. Between viruses and their respective hosts, there has always been an arms race. I would like to call it rather a race for mutations, which is represented here by the double arrows. And just as host cells have continuously evolved and refined a complex series of antiviral defense mechanisms, so viruses possess different tactics to evade such defenses. And since viruses generally replicate much faster than their hosts, what happens is that in the long run, viruses do adapt to their host, meaning that they somehow learn how to replicate and spread in the host population without massively alerting the host defenses. What has happened in the past decades is that our endless expansionism combined with globalization and all of its consequences has brought us, let's say, closer and closer to new, I like to call them ecological niches, as well as to the viruses which are associated to these niches. And this, let's say, intimate contact has enormously increased the risk of spillover, which indicates in biology it means indicates the event of basically any virus residing in a certain animal population being transmitted to a new host population, in this case the human population. And for instance, this is the case of SIV, the simian immunodeficiency virus, which was transmitted to humans uh, probably through the consumption of infected raw meat. And in humans, SIV evolved into HIV in the new host. So in the human immunodeficiency virus, here represented by the red and black. Red because HIV brings the backbone of SIV, but also carries some really unique mutations as a consequence of its adaptation to the new host. And another possible scenario concerns the evolution of the virus itself. Now, since the main weapon of viruses against the host defenses are mutations, it may happen that the presence of certain mutations, so the presence of some mutations in the viral genome can, let's say, make the virus able to replicate in another host. And this is the concept of pre-adaptation. And the pre-adaptation has been proposed as one of the putative mechanisms driving the spillover of rabies virus from bats to other animals. And now, after this 20 minutes of really basic knowledge about virology, I think it's finally time to say a few words about coronaviruses. And I purposely use the plural here because, technically speaking, the term coronavirus refers to a much larger family of viruses, which in virology is called the family of Coronaviridae. And all the members of the Coronaviridae belongs to the fourth class of Baltimore classification. So they all have their genome, which is expressed as a linear single-stranded RNA with the positive polarity. Now, one thing that I have not mentioned before, but I think deserves a few words, is that normally RNA viruses have a much higher frequency of mutations compared to their DNA, let's say, cousins. And this is because the viral workers, so the viral proteins which are responsible for generating copies of the viral genome, let's say that they work really badly. They introduce a lot of mutations when doing their job. Coronaviruses workers are, let's say, one step ahead compared to the others because they are able to recognize and fix
those errors that occurs during RNA replication. And this peculiarity has already been observed in this study published in 2004, in which scientists they had a look at the sequences at the available sequences of the SARS coronavirus, which was responsible for the epidemic that broke out in China in 2003. I mentioned before the concepts of adaptation to the host, and concerning coronaviruses, the main um, resident host in which the virus establishes reservoirs, they are called in virology. They are cattle, camels, cats, and bats. And all coronaviruses, because they are, let's say, the democratic viruses, they are transmitted via respiratory droplets. I mean, sorry, it's the other way around. They are transmitted via respiratory droplets, and therefore they are democratic viruses. So basically, any one of us can be infected with those viruses. And although I read that there are 7 strains known to infect humans, only 3 of them became quite famous. The first one is the severe acute respiratory syndrome coronavirus, which was responsible for the epidemic in China in 2003. Then there is the Middle East respiratory syndrome coronavirus, which again gave rise to an epidemic in 2012, I think. And the most famous one, which is responsible for the current pandemic, is the SARS coronavirus 2. At this point, I would like to specify one small thing, which is already highlighted here in the title. SARS-CoV-2 is the virus, while COVID-19 is the coronavirus disease 19. I want to say this because I've been reading and hearing a lot of journalists just confusing these two terms. For instance, saying that COVID-19 is spreading. No, you don't spread the disease, you spread the virus responsible for the disease. So please pay attention to this. And because it's like saying HIV and AIDS, which are two different things. You know, HIV is the virus and AIDS is the disease caused by the virus. And another important point that I wanted to draw your attention to is the fact that this virus was not created in a lab. On social media, and especially when talking to some Italian people, I really heard the most crazy, the most unbelievable, and the most ridiculous things I could have ever hear. Now, given that it's not exactly clear where does this virus come from, currently available data suggests that this virus is very likely derived from either bats or pangolins. And sequence analysis, and so for the non-virologist, the sequence represents the barcode of the virus, and the sequence is unique for each virus. The sequence analysis of the circulating coronaviruses amongst these two animals even raised the hypothesis of a potential recombination between these two viruses, which, according to this study of 2006, recombination can even occur in the case of coronaviruses. Now, what do we mean when we talk about recombination? Viral recombination is an event that occurs when two viruses infecting two different species, let's say two different animals, they co-infect. So they somehow find themselves within the very same cell. Yeah, exactly. And what happens is that the viral progeny, which will come out from this infected cell, um, will carry a mixture of information which is derived from both original viruses. And this event is thought to have facilitated the onset of the SARS coronavirus 2 spread, which, as we all have learned from the news of the past days, when this virus infected its first human, then it managed to spread very quickly and very efficiently amongst the human population. About the SARS coronavirus too, we know that this virus uses um, the ACE2 as a host receptor. In virology, you always have to see the host receptor as like the key and lock. The virus has a key to unlock its way into the cell, and the ACE2 is the target lock that the virus uses to infect our cells. And as I have shown here in the cartoon on the right, we know that this virus has a very high transmissibility between humans, between individuals. And then there are two concepts that I would like to underline, especially because I would like to say that every age is susceptible to the infection at the beginning. And in Italy, I would say still a few people believe that the elderly are the ones which are at the highest risk of infection, while they think that the young people like me are safe. No, this is not true. As I mentioned, this is a democratic virus, so literally any one of us can get it. And then a let's say worrying data for me at least is that males seems to have a much higher risk of getting severe consequences and even death upon infection of SARS-CoV-2. And I did some reading, and there are many possible explanations. And one of these, for instance, says that testosterone, so a male-specific hormones, could act as an immunosuppressor. Others says that males could have higher expression levels of the ACE2 receptor. Um, I don't know. I think that we need to investigate more to find out why males have this higher risk of death when infected with the SARS-CoV-2 virus. And yeah, since it's already half an hour of me talking, I would like to already come to the conclusions of this presentation. And what could we say? I hope I gave you the messages that viruses are obligate intracellular parasites. Um, I would like to stress on the concept that viral evolution could lead to spillover events and subsequent adaptation of the virus to the new host. Um, the SARS coronavirus 2 originated from other animals and was not an artificial virus. And I think that additional studies are needed to really understand how does this virus replicate. Because while I do agree that in the short run we need to find drugs, we need to find a vaccine or something to cure the infected patients, in the long run I think we will benefit from all the knowledge that we will get from understanding the replication cycle of this virus. And ideally we could use this knowledge to be better prepared for when the new and the next pandemic of maybe another coronavirus will come. And interestingly, however, while I try to be optimistic, I found one review in the scientific literature which was a bit concerning and worrisome for me, because this review was published in 2007, and there are these three lines in the conclusion. I hope I don't have to translate them for you, as I had to do with the Italians, but just read them. And when I read them, I realized, wow. So it means that 13 years ago, this kind of biological bomb was primed, and then in December of 2019, this bomb just exploded. So I mean, in theory, we should have not been surprised. We should have not been caught by surprise by this pandemic, because already these people told us that someone this spillover, this outbreak, would have occurred one day or the other. But yeah, I mean, I hope that we will learn something from this, and I really hope that we will be much more prepared for when the next pandemic and outbreak will occur. With this, I would just like to say thank you very much for your attention. I hope I did not confuse too much your ideas. Um, Wash your hands. That's important in spreading, in limiting the spread of the virus. If you can, stay at home. If you have any curiosity, just you can write me an email from to the address below. And yeah, take care of yourself and of your beloved ones. Bye bye.